Alright, so this begins our lecture on the endocrine system. We will kind of set up for the course is we'll look at we'll get into endocrine system and then the nervous system, the two systems that are going to coordinate. They're going to control many of the other organ systems, so everything operates. We keep things in homeostasis because we do need this communication to basically make all these cells work, make them all be in a similar function, and to keep things homeostatic. So the first of these is going to be the endocrine system, and then we'll jump into the nervous system. They're both involved in these long distance communication across the body. Endocrine system is usually a bit slower than the nervous system. So if we need something like we need our muscles to contract very quickly, right, in response to things, we'll use our nervous system. We'll get that communication and we'll be able to contract our muscles. Our nervous system can it does both electrical and chemical signal, and it is very quick. Where the endocrine system, right, if we need our muscles to contract, we can't release it from an organ, endocrine gland, and then travel through the blood and get it to contract, because that would be too slow. Okay, but it's slower, but usually the endocrine system's effects are longer in duration. Like the example with our muscle contracts and then it relaxes, so the signal goes away very quickly too. The duration of the signal for the nervous system is rather quick, where the endocrine system will have a longer term effect or longer duration of action by the hormones that they send out. All right, so let's just jump in and look at our long distance communication. Okay. Again, it's nervous system, endocrine system, and for our case in this, we have our hormones, our chemical messengers. Okay, and the signal, as we've talked about, is sent into the blood. So the endocrine cells basically send the hormones out into the interstitial fluid around them and get into the bloodstream. Okay, they will travel down. So you can see it's somewhat slower in that it has to travel through the blood, right? If you leave say your thyroid hormone gets released here, and it needs to get down to the muscles, down to the foot. And it's got to travel through the blood supply or through the blood vessels to finally get there. So it is a bit slower. It won't be instantaneous, right? But once it gets there, it can act at its target cell. And remember, with our cell signaling, what must the target cell have? The target cell must have the receptor. Okay, the cell can't be a target cell if there's no receptor, because it's not going to respond to the chemical messenger. It will not respond to the hormone. And we're a little bit different. Communication, the nervous system uses a combination of electrical signal, and that's how we're able to send the signal very rapidly. The chemical signal is just over a little synapse, so just over this little space between the neuron and the target cell. Okay, so we use a combination of both in this case for the electrical system, and that allows it to be very rapid. Where endocrines through the blood, it is a slower response by using that mechanism. And so we'll get into this. We'll get into the signaling of the neuron next in the next lectures, when we start talking and diving into the nervous system. So the nervous system uses neurotransmitters when it's talking, but there are some neurons that basically send out chemical signals in the blood, just as an endocrine cell would send signals out into the bloodstream. That's where chemicals will go. So these are termed neurohormones in the fact that they are neurons releasing the chemical signal, but instead of being across a little synapse, typically the neuron with neurotransmitters work, it is sent out in the blood, just similar to like a hormone. So these won't have a rapid effect. They'll have a slower duration of effect, these neurohormones, because they are traveled through the blood. Well, here's your basic breakdown of those long distance communication. You got when we'll be discussing the endocrine, we'll be discussing endocrine, and we will be discussing hormones or neurohormones. So hormones and neurohormones, since those are brought in the blood. In our next lectures, we'll talk nervous system. As you can see, very quick, we get this electrical signal. This gives us this very quick response, where these two, when we're put in the blood, it is a slower response. The key is that this neuron also, the difference is this neuron can only talk to this specific cells, or cells, because usually it is not one here. This will be branching off to many target cells. This mode of mechanisms with our hormones and neurohormones, essentially all tissues throughout the body have access to the hormones, or those that have blood supply have access to the hormones. Okay, but again, it is only those target cells with the receptor. Okay, but we can get this broad effect with the endocrine system, where the nervous system is a little bit more localized. So what actually makes a hormone a hormone? We've already discussed it is secreted into the blood for transport, so we can go over those long distances to communicate to those distant tissues. Okay. It's also got to act in very low concentrations, because remember, these hormones are being secreted out into the bloodstream. Okay, so they're getting diluted into this large volume of fluid, so they have to be able to act on those receptors in very low concentrations. They have to have an effect on the cells, because you can release hormones from, say, your pituitary gland, and one of the targets could be your muscles down at your big toe. So you can imagine how diluted that gets in concentration when it gets down there. So it has to have an effect with these very low concentrations. Functions of them, okay, much of growth and development. It's hormonally controlled. Metabolism, okay, regulation of internal environment, temperature, water, balance, ions. We'll be seeing a lot of this when we get into the kidneys, in hormones such as aldosterone and antidiuretic hormone, ADH. You guys are pretty aware of your reproductive hormones, your progesterone, your testosterone, your estrogen, and targets by controlling enzymatic, what are the actions at the cells? Controlling some enzymatic reactions, transport of ions across the membrane, and such as one example, aldosterone is going to cause sodium to be pumped back into the body. And since you guys know osmolarity, you know that water also falls. So that's one way we retain water. We'll see gene expression, our intracellular signalers like steroid hormones are going to aid in expression and synthesis of proteins. Okay? So they can have varied effects at the body depending on what hormone and what receptor is there on the target tissue. Alright, so major hormones. We're not going to go over in detail of all of these, but we will be doing a substantial amount. Several big players will be playing on is the hypothalamus and anterior pituitary, because these glands are going to control many of the other hormones throughout the body. We'll look a little bit at pineal gland. We'll see thyroid gland, parathyroid, a little bit of the thymus. We handle heart, because the heart, besides having endocrine cells that secrete some hormones, the heart, that is going to be a big part of our cardiovascular and how our cardiovascular runs. Liver, we'll touch a bit on these when we get to, again, cardiovascular, because we'll be looking at angiotensinogen. Stomach, seeing digestive. Pancreas we will look at, since we got our insulin and glucagon from there. We'll also look at adrenal cortex hormones, chiefly aldosterone and cortisol here. Adrenal medulla we'll handle more when we get into autonomic nervous system. We'll look at testes. Let me go back up here. Kidneys we'll look at erythropoietin. When we get into cardiovascular, look at the steroid hormones from the testes and ovaries. Okay, and we won't focus much on that. Okay, so you can see we're going to do a substantial amount. Basically, you want to know, for the most part, what endocrine gland releases what hormone, what might be some of the targets for those hormones, and what might the effect be, what might be the variable that's being controlled, or the basic effect at the different tissues. It's kind of what we'll be looking at, the hormones. We won't go into grave detail on all of these, but want to get a big picture of what's taking place with these hormones that are being released and where they are acting. So we now know what a hormone is. Let's get into the first step of how this process starts of this signaling. First, we got to get the hormones to be released. So what causes these hormones to be released from the endocrine glands or from neurons? Basically, there are three types of stimulus for the release of hormones. We got a humoral stimulus. Humoral meaning relating to the extracellular fluid. So what are the levels of certain, say, ions in this case for calcium? Another humoral stimulus would come from blood glucose in our insulin, but basically the concentrations of certain molecules in the body or in the extracellular fluid can stimulate the release of various humoral, and that is a humoral stimulus. Okay. Blood glucose is another one, right? Blood glucose is high. What's the humoral? That's a humoral stimulus to cause the release of insulin from the pancreas cells. Another stimulus is neural. Neurons can cause the release of hormones. Okay, we'll see in the posterior pituitary, neurons are releasing hormones. Here, the special case, and where these neurons are causing the release of hormones from the adrenal cortex, the release of that epinephrine, or adrenaline, as it used to be called. And then another is hormonal stimulus. There are hormones that can cause the release of other hormones. Right? We're going to see this with this combination of the hypothalamus, stimulate the anterior pituitary hormones of the hypothalamus, secreted by neurons of the hypothalamus, cause the release of hormones from the anterior pituitary, and many of the anterior pituitary hormones cause the release of hormones from other glands. So hormones can stimulate the release of other hormones in a hormonal stimulus. Okay, so we're going to see variations of this when we look at our different hormones that we're going to be looking at, and variations of these control of their release. Alright, so once released, remember the hormones get released into the blood. What are they going to go? They're going to travel down through the blood to the target tissues, and what is their function? Basically, they are going to bind to the receptors on the target tissue. Okay, as we go with our cell signaling, once bound, the target tissue can respond depending on what hormone, depending on what receptors there, depends on what response you will have at the target tissue. Because remember, there are different receptors for different chemical messengers. So you can see here a response, epinephrine, a hormone released by the adrenal medulla. Okay, you can see with an alpha receptor bound, we get vasoconstriction. With the beta 2 receptor bound, we get vasodilation. So you can see, even though it's the same hormone, we're getting a different response by what receptor is there. Okay, but that's basically the communication. We get what's the stimulus, what releases the hormone, hormones travels through the blood, and then we're going to get binding to the receptor. Okay. These hormones, okay, we want them there when we need them. When we need a certain cells to function in a certain way, we need the hormones out there. But then we have to get rid of the signal. We want it, many times we don't want the signal staying there constantly. So we have to be able to, there has to be a way to terminate the action of the hormones. Okay, one way is to limit. Okay, limit the hormone secretion. We'll see on the next slide, there's a half-life. These hormones get broken down, but when we first eat a high sugar meal, we're going to secrete insulin. Okay, that's going to be, insulin levels are high, but once we start bringing the blood sugar back down, we don't want to constantly be releasing insulin. So the pancreas start limiting their hormone secretion. That's one way to terminate the actions. Okay. And we limit the hormone secretion. They can be removed or inactive. Circulating blood, they degrade, or there are enzymes that can help break down these hormones and they can be excreted. Okay. Many times hormone activity can be terminated at the target cells. Okay. There are enzymes in many of the target cells sometimes that can break down the hormones, or they can also take in the hormone receptor complex, bring it into the cell, and then be broken down in what we call by lysosomes. Okay, endocytosis, they come in through vessels. Lysosomes merge with those vessels, and we can degrade the receptor and hormone complex. Okay, but these are just ways in which we terminate the signal. We don't have this constant signal going on. Okay. So hormone concentrations and tissue response. Okay, there's a term called half-life. That's just when does the plasma concentrations come down. Okay. Usually it's fairly quickly, not like the neuron where it's almost boom, and neuron fires, signal is taken away. Usually this can be minutes to hours, but it can also be days. So that's when you can see how giving patients hormones as treatments, many times they last for a long period of time. So you want to make sure you're giving them the proper amount. Okay, because excess amounts can have dramatic effects. You know, I still haven't figured out if this one's photoshopped or not, but man, look at those back on that guy. Okay, but you know, we can have excess amounts. Okay, you can have diseases that give you excess amounts. You can have a tumor that causes hormones to be released continuously. Okay, or some people put hormones in and give you a greater physiological dose, but you can see that ends up with, you know, this is great, you got your muscles big, but you can understand you're also doing damage to other tissues. Okay, so again, there's this homeostatic balance. Okay, we don't want to continue release and not have ability to bring down that hormone level. So we can slow it down, let time take place for the hormones to be degraded, and that can help diminish the signal, terminate the signal. Okay. So classification of the hormones, we basically can classify in two different types of categories. We can look at chemical structure. We can have polypeptide protein hormones. Of course, we need genes to make these proteins. We've already seen a couple slides in which we made insulin, which is a polypeptide hormone. Okay, so there are genes. We have to transcribe the genes, translate them, and then we can get our protein hormones. They can also be glycoprotein hormones, such as follicle stimulating hormone. Again, we'll still need a gene to make the initial protein, and then carbohydrates are added to that protein. Another class, steroid hormones. These are those that are derived from cholesterol. Okay, cortisol, testosterone, testosterone. These are all derived from cholesterol, which is a very small, if you remember, a very small nonpolar molecule. So this will allow, we'll see with the signaling and where signaling take place, steroid receptors or hormones, steroid hormones can signal inside the cell. Amino acid derived hormones, simply instead of a long protein, it is just derived from a single amino acid. Okay, histamine, thyroid hormone, these are all hormones that can be derived from a single amino acid. Histamine from tryptophan. Okay, tyrosine is the building block for our thyroid hormone. Okay, so this is by chemical structure. We can also look at actions on the target shell or target tissue. Where does the signaling take place? Okay, we can have polar versus non-polar. Okay. Our signaling for here, since it's polar, is it allowed into the cell? No, so the receptors have to be on the plasma membrane. Okay, whereas signaling such as for steroid hormones, we'll see we can use non-polar molecules because their actions can take place in the cell. The receptors can reside in the cytoplasm or the nucleus because they are small and non-polar. They can zip through the membrane. The receptor for the hormone does not have to be on that plasma membrane, since we can move through there through simple diffusion. Some terminology on hormones, prohormones and prehormones. Prohormones usually have no effect, so many peptides get translated. The initial transcript is a non-functioning hormone, but as we saw with insulin, it gets cleaved into its functional state. Okay, its actual active hormone. The prehormone would be that long chain of amino acids in which we made from the insulin gene, which is an inactive molecule. We have to be converted down, and then it gets released as insulin as the actual hormone. Prehormones, though, are actually released. Okay, released in their non-active form. Some of these we are vitamin D, testosterone. One we'll look at fairly regularly in more detail is thyroxine, T4, the thyroid hormone. Okay, T4 is a prehormone. It is not functional. It is not the active hormone. It has very little effect or no effect. The actual hormone is the T3, which it gets converted. T4 gets converted to T3 at the target tissue. Okay, so that is a prehormone. They get released in active form, and usually at some other tissue or at the actual target tissue is where they get converted into the functional active hormone. So let's look at the hormone's action by where it acts in the cell. We've seen polar hormone, right? Polar hormones cannot pass through the plasma membrane. So where must these receptors lie for these hormones? Yeah, they have to be on the plasma membrane. Okay. Just going back to our cell signaling, right? If you remember, we got our receptors. Many act through G protein couple receptors or enzyme-linked receptors. Okay, depending on the hormone, depending on the receptor, that's what we're going to get our actions either at a G protein or enzyme-linked receptor. Right? Our insulin here, binding, this is actually an enzyme-linked receptor. It's a tyrosine kinase. Binding here will activate the enzyme activity on the intracellular portion of that receptor. Okay, so summary, polar hormones can be on the plasma membrane, right, and they act through G protein couple receptor, enzyme link receptors to get that signal transduction to get a response in the target cell. This differs from, say, receptors for steroid hormones. There is our cholesterol. This is a small, very nonpolar molecule. You can see some of our adrenal cortex hormones or our reproductive hormones in our ovary. They are all basically derived from the building block of cholesterol. Okay, so that makes them very small and nonpolar. So where can these hormones receptors lie? Right? They can be nuclear, they can be in the cytoplasm, they can be in the nucleus, but they can be inside the cell. And what these receptors are going to do is they're going to act as transcription factors. There are two binding areas to these receptors, in that we have a LBD or ligand binding domain to the receptor, and we have a DBD, DNA binding domain. Okay. And these vary in that there are two families of, let's just call them intranuclear receptors. They all are going to go to the nucleus, but some lie in the cytoplasm, like our steroid family receptors. All right, and some, like thyroid hormone family, they actually are in the nucleus already. They are already bound to here, but they won't act as transcription factors until they're bound. Okay, but binding of the receptor is going to cause, basically, ultimately, for transcription to take place of certain genes. Okay. Another thing about these nonpolar hormones is they have to be transported in the blood via carrier proteins, because remember, nonpolar stuff does not like polar. And where are these hormones traveling? They're traveling in the blood in a water environment that is polar. So they have to have carrier proteins, otherwise these hormones would be all bound together because they would attract each other, being nonpolar in a polar environment such as the blood. Okay, so they have carrier proteins that help them. Once they get to the target tissue, then they can disassociate and diffuse into the cell. Okay, but they are the ligands in that they will bind to the ligand binding domain of the receptor. The other thing to see here is how many receptors. Okay, there are two receptors here. It takes two activated or bound receptors to bind and to initiate the transcription. Okay, we'll see here in this receptor here, the ligands are actually two different ligands, but when we get to the steroid family, which we're going to look at the actions and how they operate in the cell to initiate transcription, all right, they're going to have two steroid receptors bound that go in and bind to these DNA binding regions. Okay. So here's actions. Steroid hormone action. Binding of the ligand to the receptor. Here's the steroid hormone. This is the ligand binding domain. Okay. There is the DNA binding domain. So these receptors reside in the cytoplasm. the steroid hormone diffuses in to the cell, binds to its receptor, then the receptors translocate to the nucleus, because that's where the DNA that we need to transcribe is located. And there are these regions called hormone response elements on the DNA. And there are two half sites, okay, because it's going to take two bound receptors to initiate the transcription at the target gene. Okay, so we get one bound to one half site, one bound receptor to the other half site, and this is called dimerization. Di being two, dimerization of the receptors. Okay. In this case, it is a homodimer, in that the receptors are the same. On the previous slide, we saw there were two different ligands, two different receptors. Okay, it wasn't the same hormone for each receptor, and that would get you a heterodimer. The key is that they dimerize, and once bound to the DNA and dimerize, that will initiate the process of transcription. All right, so let's take this from beginning to the end for signaling with our steroid hormone, or our intracellular receptors, through our intracellular receptors. Okay, intracellular in that being the cytoplasm, or can be in the nucleus. Key is that they're both going to have similar effect in that they're going to be transcription factor. They're going to cause transcription of target genes in the DNA. Okay? But remember, our our steroid hormones, or our non-polar hormones, they are carried through the blood by protein carriers. Otherwise, they would be all glommed up together because they are non-polar in this polar blood plasma fluid. All right, so they'll travel through the blood via their protein carrier. Once they reach the target tissue, they can dissociate, and they will make their way through the plasma membrane because they are small and polar. Okay, depending on what hormone, what receptor there is, either they're going to bind in the cytoplasm. They bind in the cytoplasm, then they will translocate to the nucleus to bind to the DNA, to bind to that hormone response element region in the DNA. Okay, if it's a nuclear, they will just travel to the nucleus, bind to the receptor, and then the receptor will it may already be bound to the DNA, or will go and bind to the DNA. Okay, remember, we need to have two bound receptors that dimerize, and then we get transcription of the target gene. Essentially, these bound receptors are going to promote proteins there that are going to help initiate the transcription of that target gene. Okay, what's transcription? Transcription gives us our messenger RNA, gives us that code, so we can take it to the endoplasmic reticulum to put our protein together through translation, and then we get our new protein. Okay, so these intracellular receptors are going to act in a genomic effect. They're going to act as transcription factor to induce expression of certain genes, of certain proteins. Okay, you can see this is a bit slow effect in that we got to go through transcription, we got to go through translation to get our effect from the signaling from this hormone. So it is a longer term effect. Okay, there are some steroid receptors that are cell surface. In our lab that I worked in, we were looking at an estrogen receptor that lied on the plasma membrane. Okay, it's not intracellular, it acts at the plasma membrane, and what you see is usually through these actions you get a rapid response. Okay, our polar hormones that are going to signal through plasma membrane receptors that are on the plasma membrane are going to have a more quicker response than, say, the ones that are intracellular and working as transcription factor to give us that genomic effect, to give us that expression of proteins. Besides these hormones having their individual effect on certain tissues or regulating a certain parameter in the body, these hormones, some hormones may work together with other hormones to increase or allow the ultimate effects to take place, or they can actually work against each other, called antagonistic. So these three types of interactions, we got synergism, permissivism, and antagonism. Okay, we'll first look at synergism. Right? In synergism, we got two or more hormones interacting to give us a greater result than just the additive effect. It's not 1 plus 1, it's 1 plus 1 equals 2. It could be 1 plus 1 equals 3 or 4. We get this greater effect. A classic example of this has to do with blood regulation. Blood glucose regulation. Okay. So mechanisms are often linked together. They have overlapping effects. An example of this, like I said, blood glucose regulation, glucagon, epinephrine, and cortisol. These all have effects of increasing blood glucose levels. Okay, and you can see individually down here, okay, we have different individual effects, and you can see their effect on blood glucose level here on the y-axis. You can see individually not too great, but let's look at glucagon plus epinephrine. Okay, this is greater than just the additive effect, but let's put all three together, glucagon, epinephrine, and cortisol. All right, you can see this is much more dramatic increase in blood glucose levels than if you just had added the three individual levels down here together. So this is an example of synergism. Okay, and this, it's like 1 plus 1 plus 1 equals 6 or 7. It works a much greater effect than each individual hormone alone. Another interaction is permissivism. Okay, one hormone cannot fully exert its effect unless the second hormone is present. Okay, an example of this is the maturation of the reproductive system. Okay, two hormones must be present here in that we get thyroid hormone and reproductive. Okay, if there was just thyroid hormone alone, there would be no development. But reproductive hormones, we're going to get some development, but to get the full effect and get the full normal development and maturation of the reproductive system, besides just having the reproductive hormones there, thyroid hormone must also be present. So this is an example of permissivism. Okay. And then the final, basically in opposite direction, in that the hormones work what we call antagonistically against each other. And we've already seen this in lab and so forth, is glucose regulation. We have two hormones that are released by the pancreas. We got insulin that brings glucose down, then we have glucagon that brings glucose up. So they basically are functioning in an antagonistic manner. Usually as one, we decrease the secretion of one, the increase in secretion goes up another. So it gives us this fine-tuning effect in being able to help regulate certain variables. And so we'll start looking at the various endocrine glands or endocrine organs and the hormones they release, and we'll look at the basic or general effect throughout the body or at what target tissue they act on. And we'll start this with the hypothalamus. we'll see here that the hypothalamus can also be considered the master gland, and that it's going to regulate actually other endocrine organs to the release of their hormones. Okay, so in the hypothalamus, it has communication with both the anterior and posterior pituitary that you see here. All right, the hypothalamus will release releasing factors and inhibiting factors that are actually going to act on the cells of the anterior pituitary. And the cells of the anterior pituitary are endocrine cells. Right? So it's actually going to regulate the release of hormones from the anterior pituitary. Okay. Axons from the hypothalamus also extend down into the posterior pituitary. So the posterior pituitary is basically extension of hypothalamic axons, and you'll see they release hormones here, these neurons. So they're considered neurohormones because they're being released by a neuron. Right, so the pituitary gland, which the hypothalamus helps regulate, where we see it's two different types of tissue. The anterior is actually endocrine cells, right? And then the posterior you already saw as extension of those hypothalamic neurons. Okay. They merge together. They have the infundibulum, the stalk here, and it's almost like a little punching bag. If you remember from anatomy days, this bone region here, that's that little silica turcica, and Turkish saddle. It saddles the the pituitary gland. Okay, so the anterior pituitary. It's also the older name or previous name, adenohypophysis. Okay, hypophysis, adeno, in that it is endocrine tissue. Okay, it is of epithelial origin, and so it contains cells that are actually, there are going to be multiple hormones that are released from this anterior pituitary. But what we're going to see is, what we just talked about, is the release of hormones from here are going to be controlled by hormones that are released by the hypothalamic neurons. Okay. On the other end, the posterior pituitary, we already said, is extension of the neurons and is known as the whole name of neurohypophysis, since it is actual nervous tissue. Right? That's again, it's the axons from the hypothalamus coming down that are going to secrete neurohormones that are made in the cells up in the cell bodies in the hypothalamus there. Okay, we'll first look at the more basic of the bunch in the posterior pituitary. Okay, there will be two hormones released from here, where we'll see there's 7 released from the anterior pituitary. But the 2 neurohormones released here, we have neurons that release oxytocin and neurons that release vasopressin, or now known as antidiuretic hormone, ADH. Okay, so those are the hormones that are released from neurons in the posterior pituitary. Okay, you can see the cell bodies here, and the axons travel down to make up the posterior pituitary, and these neurons are going to release these 2 hormones into the bloodstream. So let's look at functions of the posterior pituitary hormones. We'll look at oxytocin. We've already seen oxytocin earlier in the year, and looking at contractions of the uterus when we're going into delivery to deliver the baby. If you remember, it's part of that positive feedback loop. Oxytocin increases to increase the contraction, that increases the cervical stretch, and again, more and more oxytocin gets released. Okay, oxytocin also involved in ejection of milk. Okay, this is all during pregnancy and early development of a baby. It also has roles in social, sexual, maternal behaviors. It's also known as the cuddle hormone. Okay, so when you're hanging out with and hugging with your significant other and telling them sweet nothings, you're probably secreting some oxytocin because it gives you that bonding effect. Not bounding here, but bonding. Okay, such as also with a mother and her baby. So the other posterior hormone is that vasopressin or antidiuretic hormone. You'll hear me call it as antidiuretic or ADH more often. Vasopressin is kind of the older name of it, but antidiuretic hormone. And the main function, you don't need to know the pathway here. We will get into this when we get into the urinary system, but do realize that ADH regulates water balance or water reabsorption in the body. Okay, later on we'll see this plays in osmolarity, of course, because more water, it'll be less osmolarity, or the opposite, less water, more osmolarity. So it can regulate that osmolarity in the body. Also, ADH can regulate blood pressure due to if we have more water in the blood, we can have a higher blood pressure, so forth. So that is what ADH is there. That is the function of it, is that water balance or that water reabsorption. So its target tissues are going to be at the kidneys. It's going to be the cells of the kidney to either increase or decrease water reabsorption. Alright, now let's look at the more complicated part of the anterior pituitary, because we're going to have essentially 6 hormones that are released from the anterior pituitary. And remember, these hormones are controlled by the hypothalamus. We have these, even though these cells in the anterior pituitary are endocrine, it is the release of the hormones is controlled by release of hormones from the hypothalamus, or release of neurohormones. Okay, and these hormones are being from the hypothalamus are being released into what we call the hypothalamic hypophyseal portal system. Portal system is basically 2 capillary beds in series. Typically, most tissue, we have arteries go to arterioles, bring it into the capillary bed, feeds the tissue, and then the blood comes out at the venules and up to the bigger veins, back to the heart. In this case, we have arteries, go to arterioles, bring to the capillary bed, and that would be this capillary bed here, the first one. The hypothalamus, this is where the hypothalamic neurons secrete the neurohormones. So we have this capillary bed, and then we have a portal vein, and then we have a second capillary bed. Second capillary bed is here, around the anterior pituitary, also called the hypophysis or adenohypophysis, and therefore this long name, hypothalamic hypophyseal portal system. Okay, so the hypothalamus releases these neurohormones. They travel down, and these are releasing or inhibiting hormones that are going to cause the release or inhibit the release of hormones from the anterior pituitary, from the endocrine glands, endocrine cells here. Alright, so that is the setup in this portal system and what controls the anterior pituitary. Okay, you've probably heard portal system, you've probably seen them in the digestive tract, the hepatic portal vein. Okay, that brings the nutrients that are absorbed at the stomach into the liver for processing. Okay, but this one, we got the hormones from the hypothalamus, neurohormones, released in here, go down, travel, and cause a release of hormones from the anterior pituitary. So these cells of the anterior pituitary are going to secrete hormones into the bloodstream there. All right, it is, you can see there are quite a bit of hormones. We got ACTH, TSH, okay, ACTH, sorry, adrenocorticotropic hormone. Okay, these are all hormones released by the anterior pituitary. We got growth hormone, FSH is follicle stimulating hormone, PRL, prolactin, and LH, luteinizing hormone. You can see FSH, LH have the same targets. They go to the gonads. Thyroid stimulating hormone, you can imagine where it goes, to the thyroid. And adrenocorticotropic hormone goes to the adrenal cortex. And growth hormone is going to be many tissues, and prolactin, mammillary, and other sex organs. All right, so that's the hormones. There's one missing there, melanocyte stimulating hormone, MSH. Okay, that's going to go to the melanocytes but for our sake, these are the most abundant in keeping homeostasis in the body. Okay, so you can see some of these other hormones, the hormones from the anterior pituitary, have other adrenal glands as target tissues, such as this, adrenal cortex, thyroid, okay, follicle stimulating hormone, the gonads. It's going to help produce some of those steroid hormones that get released by the gonads, okay, the ovaries or the testes. Okay. These hormones that are being released and have a target for another endocrine organ are called tropic hormones. Okay, tropic hormones have a target. The target tissue is another endocrine gland. Okay. There's actually 7 with the MSH, the melanocyte stimulating hormone, MSH. Okay, but we're going to deal chiefly with these 6 here. As we said, those hypothalamic neurohormones are going to control. These hormones, neurohormones, are releasing or inhibiting hormones that are going to direct the cells of the anterior pituitary whether to secrete hormone or not. Okay. Again, they are tropic. So here is the setup of all the hormones and through the hypothalamus hormones. Those are those inhibiting and releasing hormones that are going to go act to the anterior pituitary. And then here are the hormones of the anterior pituitary and what their actual targets are. Okay, you see dopamine, neurotransmitter. It's released into the hypothalamus or to the portal system and actually inhibits the prolactin release. TRH, thyrotropin releasing hormone, goes to the anterior pituitary, causes a release of TSH, and so you can see this flow of hormones. Just like many factors in the body, we try to keep homeostatic many of our hormones. We need the right amount to make sure the cells are operating properly and we're getting proper functioning, but we don't want too much and we don't want too little. So what do we need in that case? How do we control it to keep it in this homeostatic range? What keeps us there? Yeah, our negative feedback. So these loops, you'll hear it as a hypothalamic you'll hear it as an HP, say HPT axis. Okay, H here, P T axis, hypothalamus, pituitary, thyroid axis. So this pathway is going to be subjected to negative feedback. Okay. So let's look at our feedback. We'll go through our feedback and how some of the control of these hormones is made, and then we'll start going into the different endocrine glands. Okay. Remember our stimulus, in this case is our hormones can go and shut off the response. Looking at this, let's look at the negative feedback through the HPT axis, HPT, all right, and so T3, T4, those are your thyroid hormones. They are good. You know, we need a certain amount to keep our basal metabolic rate going and so forth, but we don't want too high, too low. So how do we get it? This is what's going to control our feedback, the levels of T3, T4. When they get high, they can go to the anterior pituitary to inhibit the release of TSH, or go to the hypothalamus to inhibit the release of TRH. Okay, so there's our negative feedback. When they're low, there's not much negative feedback, and there will be higher amounts of TRH, TSH, and you'll get higher amounts of release of T3, T4. But once they get in that homeostatic range, they start creeping up to the high level, they can shut it back off through the negative feedback. Okay, so that's going to control the release of these hormones. Okay. The reproductive hormones, okay, estrogen, testosterone, the steroids, okay, the release of those is going to be controlled by a combination of LH and FSH from the anterior pituitary. Okay. There's actually a short and long loop feedback mechanism for the release of these hormones. Okay, the steroid hormones, again, they're the final product, the end product, and we want the levels. We need a certain amount, but not too high, not too low. Say levels are getting high, we can do what's the long feedback. Long feedback is with the final hormone from the testes. Okay, either estrogen, progesterone, or testosterone in males. We can feed back, and that can inhibit the release of GnRH, gonadotropin releasing hormone, from the hypothalamus, or it can feed back and inhibit the release of LH and FSH. Okay, there in this loop there is a short loop of negative feedback, right? Instead of the hormones down here shutting off the release, FSH, LH can act on the hypothalamus to inhibit the release of GnRH. Okay, so there's two negative feedback loops taking place here. There's long with the hormones secreted by the gonads. They can go down and shut down or inhibit the release of hypothalamic hormones or the anterior pituitary hormones, or the short loop, those anterior pituitary hormones can inhibit the release of the hormone, the gonadotropin releasing hormone, from the hypothalamus. Okay, the loop for thyroid doesn't have that, so it depends on the hormone, depends on whether we see negative long loop feedback and also short loop feedback. Not all hormones have that feedback. Okay. All right, so let's start going in. We'll look at the different hormones. We'll jump back a little bit, go back a little bit. We'll look at our prolactin and growth hormone. Okay, you'll see prolactin. dopamine actually inhibits this is inhibition. Okay. Again, it's going to control milk production. It also has some immune system regulation in both sexes, but for females during breastfeeding time, it is controlling the milk production. Okay. You'll see, besides prolactin releasing factors, there's also TRH. That's for the thyroid though, right? TRH actually has some influence on prolactin to increase milk production. Okay, growth hormone. You got growth hormone releasing hormone. Wonder what it does. Yeah, it causes the release of growth hormone from the anterior pituitary. There is an inhibiting factor, somatostatin, that will cause the inhibition of GH, the growth hormone. Okay. And most people know or have heard of growth hormone. It's going to affect the metabolism. It's also going to stimulate IGF, insulin-like growth factor from the liver, which will go down and cause growth of many tissues. So this has a response at many target tissues. So growth hormone release is going to affect many tissues throughout the body, just certain individual ones. So now the adrenal gland, similar to the the pituitary, it has two sections. There's one that is more neural based, and there's a section that is endocrine based or epithelial derived. Okay. And so we got the adrenal cortex is the outer portion. We're going to have those typical endocrine tissue, and there's going to be multiple hormones that are being released from the adrenal cortex. Okay, it's going to secrete steroid hormones, hormones that are derived from cholesterol. Adrenal medulla probably looks familiar to some from your anatomy days when you went over the autonomic nervous system, okay, because there are neuroendocrine cells, okay, chromaffin cells residing in here. All right, and those neurons of the sympathetic nervous system come in here to cause the release of epinephrine. Okay, so it is neurohormones being released from the adrenal medulla at intersection. Okay. So looking at the more complex part of we're looking at the cortex here. You can see we get multiple hormones being released from this adrenal cortex, and it's separated into three sections: zona glomerulosa, zona fasciculata, and zona reticularis. And each of these regions are going to secrete different hormones. The zona glomerularis is going to secrete mineralocorticoids. Okay, remember these are all derived from cholesterol as a beginning product. Aldosterone, we're going to see very similar what we saw with ADH. It's going to be retention of sodium, and when sodium gets retained, again, water follows it. Remember our non-penetrant, this is where a bit of our osmosis comes in. We'll get into it quite a bit more when we get into blood pressure and we get into the kidneys, the urinary system. Okay, the middle, the bigger section, zona fasciculata, okay, releases cortisol. Many of you know it as kind of your stress hormone. Okay, corticosterone is another one. But this is where that adrenocorticotropin hormone from the anterior pituitary, this is the target for the cells that release cortisol. Okay, it's going to stimulate protein degradation, gluconeogenesis, lipolysis, and also, you've heard of steroid suppressing inflammation. They give you artificial cortisol, and that will suppress the immune system for certain disorders, for disorders, autoimmune disorders. Many times they suppress the immune system because the immune system is attacking your own cells. Okay, but again, this is where that target of ACTH is for cortisol release. Okay, we also get androgens. Okay, those steroid hormones, those hormones we get from the gonads. There is superficial amounts released here from the reticularis. The bulk of those hormones, those reproductive hormones, are secreted, or the androgens are secreted by the gonads, but there is a small amount also secreted by the adrenal cortex. Right. You can see our synthesis pathways. It all begins, steroids begin with cholesterol. Okay, and depending on all these pathways, you can see progesterone ultimately ends up in aldosterone. So there's a middle intermediate here. So progesterone is an intermediate for making corticosterone and cortisol. So depending on what hormone the cell releases, all these are enzymatically made, and so they would have various enzymes to create the different products here. Okay, you can see cortisol, aldosterone up here, up in the zona glomerulus, right, and then those androgens down here in more of the zona reticularis. Okay, but again, they are steroid hormones, they're cholesterol, so they are able to zip through the membrane. Remember, but they have to have a carrier protein when dissolved in the blood, otherwise they will not dissolve and they would all glom together. And you'll see, unlike some hormones, like your peptide hormones, can be made and stored in vesicles. Can these be stored in vesicles? No, they can't be stored in vesicles because they can zip right out through the membrane. So many times our steroid hormones are made on demand because they can just make their way out anytime. We can't store them in vesicles because of their lipid solubility. All right, so let's look at the control. Since ACTH controls cortisol release, okay, our CRH from our hypothalamus, that's our H. A causes the release of ACTH from the pituitary, and then the ACTH goes to the adrenal cortex, the A, and causes the release of cortisol. Okay, similar to the reproductive hormones, you can see that there is a long loop, negative feedback loop, and a short negative feedback loop. Okay. So next in line, we can look at the thyroid gland, and we're actually going to see there's the thyroid gland and the parathyroid gland. But let's first focus on the thyroid gland. And there are two hormones that are going to be released from here. And you can see kind of the setup. There are these follicles, and they're going to be filled with basically precursors to the thyroid hormone. So these cells that line the follicles, the colloid follicles, okay, and the precursors, that thyroid globulin, are the follicular cells. So these cells that line these follicles are the follicular cells, and they are going to take the precursor from here. When stimulated by TSH, they're going to make thyroid hormone. Thyroid hormone is T4 and T3. All right, that is the thyroid hormone. This, if you remember from our earlier slides, this is a pre-hormone. Inactive, this is the active form, the T3. Right? Besides the follicular cells, these little blue cells, which are the parafollicular cells. Okay, and they're going to secrete a hormone called calcitonin. Right? This causes secretion of calcium, therefore reduces calcium level. I like to think of calcium level with the calcitonin, with the C, as cut down calcium levels in the blood. Okay, because we're going to see there's parathyroid hormone, which actually works antagonistically to this calcitonin. Okay, but follicular cells are going to make the actual thyroid hormone, the T3, T4, and the parafollicular cells are going to secrete the calcitonin, which the function is to cut calcium in the blood. Okay, we want to keep calcium regulated, calcium levels, concentrations in the plasma, blood plasma, in a homeostatic range, and calcitonin is a way we can reduce calcium in the blood. Okay. What are the main functions of thyroid? The main function that it's known for is setting the basal metabolic rate. Okay, it has some influences on those macronutrients. Okay, another main is increased heart rate and force of contraction, and we've seen it in normal growth. Adding growth hormone needs to be available for that permissiveness of growth hormone to be able to exert its full growth effects during those windows. Okay. So normal development of the nervous system. It's another function. If it is missing during certain growth periods in young babies, then you will lose the normal growth of the nervous system. Okay, but as adults, one of the main big function is it is one of the main players in your basal metabolic rate. Okay, so if you have an excess of thyroid hormone, you're actually going to have a quick basal metabolic rate, and you'd be able to burn calories much quicker. If it's low, you're going to have a low basal metabolic rate, and you may put on some weight because you have this lower basal metabolic rate. Alright, so looking at the thyroid hormone, it's basically made from two tyrosine molecules, moieties here, them together, and you can see thyroxine, T4. T4, and what is the 4? The 4 stands for the fact that there are this I atom, that is your iodine. Okay, and there are 4 iodine atoms for T4. Okay, that's the pre-hormone. When we get to the cells, this gets to the cell, all right, this iodine is going to be removed, and you get triiodothyronine, T3. See why we call it T3, right? Okay, but now we just have 3, so the number has to do with how many iodine are attached to the thyroid hormone. Okay, that is the active form. So to be able to form active thyroid hormone, there has to be iodine in the diet so we can put it into the thyroid hormone. Okay. Some of the effects, say if you have low thyroid, you have low functional thyroid hormone, okay, you can see some of the adverse effects that can take place. Okay, be cold, be slow and kind of sleepy, lethargic. Okay. Might have slow pulse, wouldn't perspire. Some of the effects. The opposite end, hyperthyroidism. Okay, excess thyroid hormone, functional thyroid hormone. You have kind of jitteriness,
Okay, you'd be hot all the time, and cause you to have excessive perspiration, so forth. And you have nervousness and jitteriness. Okay, so those would be some effects of having either low or high thyroid hormone in the blood. Okay, let's look at some disorders in which thyroid hormone can take place, and think in the terms of negative feedback. How do we get negative feedback? If you remember iodine is essential to make functional thyroid hormone. If you don't get iodine in your diet, right, you cannot make functional thyroid hormone. So you would be hypothyroid. You wouldn't have the activity of the thyroid hormone at the target tissues. Okay, the problem is what's going to happen to negative feedback. Remember that thyroid hormone goes back to shut down TRH and TSH. Okay. One thing about TSH, certain hormones are known to be what we call trophic, in that they cause their target tissues to grow. Growth hormone would be a trophic hormone, and it causes the tissue to grow in size. All right, TSH is trophic. So if there's excess TSH, the hormone or the thyroid is going to grow. And you can see when it grows, we can get, right, a goiter. That is from too much TSH activity, too much stimulation of the TSH receptor on the thyroid gland. Right, so let's look. Hypothyroidism. Okay, again, we already saw that slide with all the effects that might take place, but why might we get this goiter with hypothyroidism? Let's at the iodine deficiency. If iodine is deficient, we would get the effects here, but we could also get a goiter, the growth of the thyroid. And why would that take place? Remember our T3, T4. They will have effects on the target tissues, but also they are used for the negative feedback. They shut down the release of TRH and TSH through the negative feedback. Okay, to negatively feedback though, the hormone has to be the functional hormone with iodine. Without the iodine, this negative feedback is not taking place, and so what happens to the levels of TRH? They are not inhibited by the negative feedback, and they increase. Levels of TRH increase. If TRH increases, it causes more release of TSH, and you have an increase of TSH. And remember this TSH. It is a trophic hormone in that it will cause the growth the size of the thyroid, right? And so that is what's taking place here as we get growth of the thyroid. On the opposite end of the spectrum, we could have Graves' disease, which would give us hyperthyroidism, excess thyroid, but we'll also see that you can still get a goiter even though you have iodine in the diet. You have actually excess thyroid hormone now, functional thyroid hormone. So you'll get the effects of this at T3, T4. You'll be jittery, you'll be sweaty, you'll be hot all the time because you have excess thyroid hormone. Plus, you will have this happening. There will be lots of negative feedback. So that would actually decrease the TRH and TSH, right? Which is the opposite of what we saw with the iodine effect, with the low iodine. Okay, but why does we still get a goiter? It's because in Graves' disease, it is an autoimmune disease. All right, your immune system is great in that it makes these antibodies that go and attach to foreign antigens and allows us to detect them and allows us to get rid of them. Problem in this case with Graves' disease is this antibody that was created actually stimulates the TSH receptor. It's an agonist to the TSH receptor. And the problem is the immune system is not affected by this negative feedback. So it is constantly making these antibodies, and these antibodies are constantly stimulating the thyroid or the TSH receptor and causing the thyroid to grow, along with causing the increased release of this. That gives you the effects of hyperthyroidism, the sweatiness, the hot all the time, the rapid heartbeat, jitteriness. Okay, so in both cases you can have hyperthyroidism and you can have hypothyroidism, and you can get goiters in both, but doing from different mechanisms. So thyroid releases thyroid hormone, T3, T4, and calcitonin, which cuts calcium, lowers calcium levels in the blood. The antagonist to that calcitonin is this parathyroid hormone. And these parathyroid gland, they are 4 little clusters of cells on the posterior side of the thyroid gland, right? And they have these receptors that respond to calcium. When calcium is low, these receptors aren't being stimulated, and that will stimulate the parathyroid hormone to be released. Okay? And this has the effect of increasing calcium. Okay, bone reabsorption. Okay, bone's getting broken down. Remember, bone is a big store of our calcium. When we break down the bone, it gets released into the blood. Okay, we can cause reabsorption of calcium, and the intestine can increase the absorption of calcium. So we have all these effects that go to do the opposite of calcitonin, in that parathyroid hormone increases blood calcium levels. Okay, purge. I like to say purge. You're going to purge calcium into the blood. For the P, calcitonin's cut it down. P, parathyroids to purge calcium into the blood with the P. So now we'll look at the hormones of the pancreas, and there are multiple hormones, but the two we'll focus on are glucagon and insulin. Right, and you've seen pancreas probably from your anatomy days. You remember it's also entailed in most of the organ is actually exocrine organ. It's going to secrete all those enzymes into the digestive tract for digestion. But there are little islets along Langerhans. These cells of the islets along Langerhans, there's alpha cell and beta cells. Right, beta cells secreting the insulin, and alpha cells secreting the glucagon. And both of these are going to be involved in the regulation of blood glucose. Glucagon working to bring blood glucose up, where insulin works to bring blood glucose down. Hey, glucagon is not alone. There are multiple hormones, such as epinephrine, cortisol, and growth hormone. As we saw with that synergism, those hormones all go to increase blood glucose. So there's multiple ways, multiple pathways into increasing blood glucose, but there is only one way to decrease it, and that is through insulin. Okay? Insulin is going to tell the liver, tell the skeletal muscle, tell the adipose cells to bring blood glucose in. It's going to allow blood or glucose to go from the blood into those cells, and if they're in the cells, then they are removed from the blood. Okay, where glucagon, on the other hand, its effects are going to be to increase blood glucose, and it's going to tell the liver has stores of what we call glycogen. Remember, glycogen, those are long strands of glucose, tell the liver to break down and release glycogen, or break down glycogen to release glucose into the blood, as well as tell the liver to also make glucose from other products or other substrates. Okay, so they're working and what we call antagonistically. One's raising blood glucose, the other is bringing blood glucose down. So, since insulin, we've all heard of diabetes, and that is a disorder in the signaling of insulin. At least diabetes 2 is a disorder in signaling insulin. And what is the effect? What is the mechanism in which insulin brings blood glucose down? So, since we've done our chemical signaling, right? Insulin binds to its receptor. This could be on the liver cells, okay, the skeletal muscle, the adipose tissue, but when it binds to its receptor, what it is causing is we get the tyrosine kinase, so it causes phosphorylation, but ultimately the signaling results in these Pac-Man things here are GLUT4 transporters. They transport glucose. They do facilitated diffusion, right? Glucose is high outside and low inside. Glucose can move through these transporters. Remember, glucose can't do simple diffusion and move directly through the membrane. All right? So what we're saying is when insulin's not around, there are very few of these on here. Okay, what the insulin signaling is causing for these to be inserted onto the plasma membrane of those cells of the target tissue. Okay, without those transporters there, glucose cannot move in. But when we eat a bag of Skittles and glucose is high, insulin gets released, and if all is working well, this signaling from insulin will cause these to go onto the membrane. The glucose can come into the cells, and now we have decreased the glucose in the bloodstream. Okay. Insulin's other effects, right? It will cause glycogen synthase, stimulate the enzyme because now we have glucose in the cell. Now we can store it away as glycogen if need be. Unfortunately, there is only a certain amount of glycogen that can be stored, and so the rest gets. Insulin also influences or promotes lipogenesis. All right, the creation of lipids and fats, so can be stored away as fat also. Okay, the main function is it causes translocation of these to be inserted on the membrane, and now we can get glucose into the cell. If this signaling, such as what happens in diabetes, is not working properly and these transporters don't get translocated to the cells aren't going to take in the blood or glucose from the blood, and what happens is the glucose stays out in the blood, and so we get in diabetes we see raised levels of blood glucose. So here's the antagonistic action of these two hormones. How do we keep the blood glucose homeostatic? All right, we eat a bag of Skittles, blood glucose goes up. All right, that's going to cause glucagon to go down because we don't need to add more glucose to the blood. We need to get rid of it, and that will cause the beta cells to secrete insulin. If insulin signaling is working, we get cellular uptake of glucose, and blood glucose levels will go down. On the opposite end, if you've been fasting for days and your blood glucose levels start to dip, right, that's when we want glucagon on board. Those alpha cells will increase glucagon secretion, and the beta cells will decrease insulin secretion. Okay, now we don't have insulin, so this isn't taking place, and we can do our glycogenolysis from our liver. Our liver can break down glycogen and put glucose in the blood. The liver can also do what's called gluconeogenesis. Right? You've seen that term before, in that it can make glucose out of non-carbohydrate molecules. Both of these effects increase plasma glucose or blood glucose, and now we've counteracted the stimulus. We started with low glucose, now blood glucose, now we have increased it. Okay, but you can see these hormones are working in opposite direction, working antagonistically. Okay, there's the feedback loop on the insulin. Okay, blood glucose intake, that will shut it down. All right. Another gland up in the brain. Okay, near the what we call the epithalamus, gland here. Okay, pineal gland, its function is to secrete melatonin. Okay, it's going to receive information from what we call the suprachiasmatic nucleus, that houses the biological clock. Right, light's going to inhibit the neurons in the SCN, and then the SCNs aren't going to project back to the melatonin and tell them to release melatonin, where when it is dark outside, there's less stimulation, and that's going to cause, actually, dark is going to cause stimulation of the SCN, and the SCN is going to tell the pineal gland to release melatonin. Okay, so that's where we get, when you dark outside, this is where they've been talking about with all our technology with the bright blue light in it. That blue light inhibits those neurons in the SCN. Okay, you've heard, don't read your phone, don't use your tablets at night. I have a Kindle that I read on, and it's kind of nice because it has a spot where you can turn off the blue light, so it becomes really pale, so it won't inhibit these neurons here, okay, or inhibit them less, and therefore I can go to sleep. Whereas if you're looking at those bright phones, bright tablets, that will inhibit the SCN, and that will inhibit the release of melatonin. Melatonin is there to cause you to be sleepy and put you to bed. All right. All right. Last but not least, we will look at the reproductive hormones, and we will only touch base on these because we will see them later in the semester when we do reproduction. But remember our control. We got the gonadotropin releasing hormone from the hypothalamus, causes the release of LH and FSH, luteinizing hormone and follicle stimulating hormone from the anterior pituitary, which go down and have effect on the gonads of both the male and the female. Okay. The gonads. Okay. Testosterone. These cells respond to LH and FSH, the Leydig cells of the testes here, which results in the stimulation and release of testosterone. Estrogen, we have theca and granulosa cells of here in the ovaries. These are responsible for the production of estrogen and the precursor androstenedione. Okay, so these are the hormones that are going to secrete, or these are the glands, the testes or the ovaries, that are going to secrete these reproductive hormones, or sex hormones as they're known as. There is a temporary in females. The placenta is an endocrine organ. It's going to secrete progesterone, estrogen, chorionic gonadotropin, right? HCG. So this is temporary organ, temporary endocrine organ. Since after birth it is delivered with the baby, and therefore you lose the endocrine effects. This, since it is a temporary, and it's one of the few things that make this human chorionic gonadotropin hormone, this is what those pregnancy tests that you're peeing on are searching for. They are reacting to this, since it is only in high levels when the placenta is being established. Okay. So looking at the steroid production, you see the pathways again they are steroids, so they are first derived from cholesterol, again small and lipid soluble, all right, lipophilic, and nonpolar, and therefore it can zip through the membrane. These steroid hormones can zip through the membrane. Remember, many of the receptors are intracellular. They're either in the cytoplasm or the nucleus, and from this you don't need to memorize all of these enzymes. What I'm just showing you is that depending on what enzymes the certain cells have depends on what hormones get produced. Okay, and so you can see our aldosterone up there. That's our aldosterone from our adrenal gland, but for our sake on this end, we're going to be making estradiol. Okay, you can see testosterone gets made. Testosterone can actually be as a precursor to making of estrogen. You have to take testosterone a couple more steps to get to estradiol. Right. So that is it for endocrine. Again, some of these hormones we will see again in other organ systems. We definitely will see aldosterone. We will see ADH, because they are going to be big players in the urinary system and also in the cardiovascular. Right? These steroid hormones, these sex hormones, we'll see in reproductive. So we'll be touching base again. For our purposes, you need to know general function of the hormones that I've gone over, and that is it. We will be moving on to the nervous system next, the other long distance communication system, and there we got to go over the electrical signaling along with the release of the chemical messengers, the neurotransmitters.